Welcome to one of the most fascinating stories in evolutionary biology. Today, we are going to explore the succession of the kidney in vertebrates. Imagine upgrading a simple water filter in a small cabin to a massive, high-tech water recycling plant in a desert mansion. This exact process of upgrading is what happened inside the bodies of vertebrates over millions of years of evolution. We will discover how animals adapted their internal plumbing to survive in increasingly challenging environments. To begin our journey, let us think about why an upgrade was even necessary. If you live in a small cabin, a basic water filter attached to your sink is perfectly fine. But as you move to a massive mansion in a dry desert, you must build an incredibly advanced, highly efficient water recycling plant that saves every single drop of water. In comparative anatomy, this biological upgrade process is called the succession of the kidney. It is the story of how vertebrates evolved their excretory systems to handle the transition from water to land, ensuring their survival. Now that we understand the overarching theme of this evolution, let us look at the formal biological definition. In comparative anatomy, the succession of the kidney refers to the developmental and evolutionary sequence of the vertebrate excretory system. Every kidney develops from a specific embryonic tissue known as the nephrogenic mesoderm. According to the accepted biological model, this remarkable tissue gives rise to three distinct types of kidneys. Crucially, these kidneys develop in a strict front-to-back, or anterior-to-posterior, sequence along the body of the developing embryo. Following this front-to-back developmental sequence, the very first structure to appear is the pronephros. This is the most primitive stage of the vertebrate kidney. Because it forms at the very front, or anterior of the body, it is often called the head kidney. While it is an important developmental stepping stone for all vertebrates, it is only fully functional in certain larval fishes and amphibians. For these simple aquatic creatures, the pronephros does exactly what it needs to do, but it is far too basic to support larger or more complex life forms. Moving further down the embryonic body, we arrive at the second stage of kidney development, known as the mesonephros. As the name suggests, this structure forms in the middle of the body. For many aquatic animals, evolution stops here. The mesonephros serves as the permanent adult kidney for fishes and amphibians. It is a significant upgrade from the pronephros, capable of handling the metabolic waste of fully grown aquatic vertebrates. However, because these animals live in water, the mesonephros does not need to be particularly good at conserving fluid. This brings us to the final and most advanced stage of our evolutionary sequence, the metanephros. Forming at the posterior, or back, of the body, this is the highly complex adult kidney found in reptiles, birds, and mammals. The metanephros is an absolute marvel of biological engineering. It introduces advanced microscopic structures, such as the loop of Henle, which act as powerful water-saving devices. Without this specific evolutionary upgrade, life on dry land would be absolutely impossible for vertebrates, as they would rapidly dehydrate and perish in the terrestrial environment. Before we look at the mathematics of how this works, let us emphasize why this succession matters so much. The primitive pronephros and mesonephros are perfectly fine for animals that live in water. They can afford to lose a lot of water while flushing out toxins. However, when animals moved to land, water became a precious, scarce resource that had to be conserved at all costs. If human beings still had a mesonephros instead of a metanephros, our bodies would be so inefficient at saving water that we would have to drink dozens of liters every single day just to survive. To truly understand why the metanephros is such a massive upgrade, we need to look at the fundamental mathematics of renal function. The primary job of any kidney is to filter the blood, save the good substances, and excrete the toxic waste. This is represented by the equation E equals F minus R plus S. E is the final excretion rate, or the actual urine produced. F stands for the filtration rate, the initial fluid squeezed from the blood. R is the reabsorption rate, the crucial amount of water saved and pulled back into the body. Finally, S is the secretion rate, accounting for extra toxins pumped out at the last minute. When the reabsorption variable R increases, the final excretion variable E decreases, meaning the animal conserves much more water. The shift to the metanephros is essentially a biological mechanism to maximize the value of R. Now that we have our formula, let us see this with a concrete example of a primitive freshwater fish using a mesonephros. We are given that its kidney filters fluid at a rate of 100 milliliters per day. It only reabsorbs water at a rate of 20 milliliters per day and secretes additional toxins at 5 milliliters per day. We need to calculate the final excretion rate. We choose our fundamental excretion equation because it perfectly models this fluid balance. By substituting our given values, we take the initial 100 milliliters, subtract the 20 milliliters saved, and add the 5 milliliters of secreted waste. This gives us a final excretion rate of 85 milliliters per day. Out of 100 milliliters filtered, a massive 85 milliliters is lost as urine, which is fine for a fish surrounded by fresh water, but disastrous on land. Following that aquatic example, let us now look at a terrestrial mammal, like a desert mouse, which possesses a highly advanced metanephros. This animal filters fluid at a massive rate of 1,000 milliliters per day to thoroughly clean its blood. Thanks to advanced structures, it reabsorbs water at an incredible rate of 990 milliliters per day, while secreting toxins at 10 milliliters per day. We apply the exact same excretion equation to see the mathematical difference. We start with the 1,000 milliliters filtered, subtract the massive 990 milliliters rescued by the kidney, and add the 10 milliliters of secreted toxins. The final excretion rate is just 20 milliliters per day. Even though the mammal filtered 10 times more blood than the fish, it only lost a tiny fraction of that water, proving why the metanephros is essential for terrestrial survival. Now that we have calculated the incredible efficiency of the advanced kidney, let us look at a diagram showing exactly how these stages are arranged. On the screen, you can see the embryonic body axis, with the anterior, or front, on the left, and the posterior, or rear, on the right. The large arrow indicates the developmental and evolutionary progression. At the top, in the anterior region, we see the simple pronephros with its basic tubules. Moving down to the mid-body region, we see the mesonephros, which has a larger number of tubules. Finally, at the bottom, in the posterior region, we see the metanephros. Notice how incredibly dense and complex the metanephric tubules are compared to the earlier stages. This structural complexity is exactly what allows for the massive water reabsorption we just calculated. To wrap up our journey, let us review the most important ideas we have covered today. First, the succession of the kidney is a beautiful example of biological problem solving, occurring in a strict front-to-back sequence along the embryo. Second, vertebrates upgraded from a simple pronephros to a mid-body mesonephros, and finally to the highly complex metanephros. Third, this entire evolutionary progression was driven by the need to transition from water to land, which required extreme water conservation. Finally, as we saw in our calculations, the advanced metanephros achieves this by maximizing the reabsorption rate, ensuring that precious water is kept inside the body. You have now mastered the anatomy and mathematics of kidney succession and can fully appreciate how your own kidneys keep you alive every single day.